So let's begin a review of the 2026 AP Bio FRQ. These are the Mr. Grodski solutions, and of course I do not work for, nor am I endorsed by, or affiliated with anybody from the College Board. These are just my own personal interpretations of the problems. My solutions are my own, and they represent what I think are the correct answers. The College Board may disagree, so let's be crazy and start in order with number 1. Question 1, part A, gets right to it. It says describe the 3 structural components of a nucleotide, and they were not specific whether this was going to be RNA or DNA. Now I drew a DNA nucleotide because I have a deoxyribose here, but the 3 parts I needed was a phosphate, a pentose sugar. You certainly could say 5-carbon sugar. They might be able to accept just a sugar, and then a nitrogenous base, which of course is the coding language of life. That's an A, T, C, G, or U if we're using RNA. So again, they were not specific. So again, phosphate, sugar, nitrogenous base should be accepted. And this has something to do with the experiment that they're asking you to analyze and answer questions to in part 1, which by the way, I thought was very, very fair. Okay, but again, just a knowledge-based question. So let's get into this study a little bit. If we were doing the test, I would tell my students to definitely go to the questions first. But since we're trying to understand this question as a whole, let's just go right after the most important parts of this. So number 1 says molecules known as dinucleotide polyphosphates type of a nucleotide are signaling molecules that accumulate in plant cells during dry or stressful conditions. So these are going to be our chemical messengers in this problem, right? So there's some kind of nucleotide polyphosphates that are going to be the chemical messengers. Now, what are they going to signal? Let's continue reading. Here we go with stomata. Right down here are pores on the surface of leaves that open when exposed to light and regulate the flow of carbon dioxide in and out. We should know that already. In response to dry conditions, many plants close their stomata, which reduces water loss. Things that we probably should have learned in our course already. They're very nice to tell you that. Now, here comes the meat and potatoes of this question. Scientists hypothesize that the dinucleotide phosphates AP4A and CP4C bind to a DORN1 receptor. Okay, now that's going to be the actual protein receptor that's going to accept these signaling molecules, right? And they're hypothesizing that these chemical signals bind to this receptor and somehow initiate a signaling cascade. Boy, that should really resonate with you in terms of the phosphorylation cascade, that domino effect that leads to some sort of cell response. And there we have it, stomata to close. So we've got ourselves a signal transduction pathway here where we have the signaling molecules that lead to a cell response. Okay, so that's been set up. Scientists expose the plants to light to open the stomata and then place samples of leaves in a buffer alone or in a buffer containing these 3 compounds. These 2 are the nucleotide polyphosphates, and then we have something called ABA, which is a molecule that causes stomata to close. That's independent of what they're trying to study here. Okay. The scientists measure the size of the stomata each sample and calculate their sizes. Okay, so all this is good information, and we've got our study here. We've got our data, I should say. Okay, and we're looking at the effect of these nucleotides here on a DORN receptor, or at least we're looking at the nucleotides and their effect to what? Change the size, close the stomata. Okay, so let's go on to question 1. Question 1, part B, identify the dependent variable in the scientist's first experiment. So if we do that, the dependent variable is something we're going to measure, my friends. So what we measure is normally on the y-axis to see how tall these bar graphs are going. So the dependent variable we're measuring here is the relative size of the stomata. How big are these stomata? Are they wide open or are they closing? And again, if you're not sure about that, reread what the hypothesis, or what the scientists were trying to hypothesize. They're trying to hypothesize that these 2 chemicals, which are chemical signals that are a type of a nucleotide, bind to a certain receptor and at some point they're going to cause the stomata to close. So the closing of the stomata depends upon those chemical signals. Either way, you should get that down. That's the dependent variable. All right, so that should be straightforward. And if you got that wrong, always think of dependent variables are things that we measure. And again, also the other thing you can tell yourself is what depends on what? Does the signaling molecule depend on the closing of the stomata, or is the closing of the stomata dependent upon the treatment groups? And it's clear it depends upon the type of treatment groups as the data is presented. So on to part BII. Part BII, based on figure 1, describe, now we need more information with this action verb, and describe the relative size of stomata in the CP4C treatment group as compared to the size of the stomata in the group treated with the buffer alone. Okay, very simple. Now, important when you do this, if error bars are given to us and they're telling us the error bars are plus or minus the standard error of the mean, and they're being consistent from last year's test where they're not doing plus or minus 2, so we're going to have to assume that we're going to use the error bars at plus or minus 1 standard error of the mean to be something we can measure reliability or confidence in. So they said CP4 compared to the buffer alone. And clearly we can see that the error bars do not overlap. So there is a definite decrease in the relative size of the stomata as compared to the buffer. I like to make sure that the AP readers know that I know that there is error bars there. By having my students write, there's a significant decrease. That's the word I would ask my students to write. Without significant, you may think that the error bars have no meaning. So of course they do. It's a measure of confidence, right? So therefore they're not overlapping. Therefore there is clearly a difference here. So that's the answer. So we would say there's a significant decrease in the size of the stomata in the CP4C treatment group as compared to the buffer solution. I definitely make my students write down. Part BII, justify the use of buffer alone as a control in the scientist's experiment. So the buffer alone, my friends, is an important negative control because we don't have any decreasing going on with no chemical signaling. So you can see that this value of size of stomata is the largest. These openings are the largest. They're not closing. They're not being affected by any chemical messengers or any other chemicals to close that stomata. Remember, they're in light, which they normally are open to exchange the CO2 and O2. The reason for the buffer is to act as a control to measure if the treatment groups have an effect on the plant's stomata. You can also reason to have this control is to eliminate other variables. There's always two reasons why we have a control that I always tell my students. Number one is to be a value to compare the other treatment groups to see if there is a significant difference, to see if there is an effect by the treatment groups compared to the control that doesn't have the treatment. And also we want to eliminate other variables that might be leaking into this study. So if we want to eliminate, let's say the buffer, maybe the buffer could be causing the opening and closing. So to eliminate the buffer as a potential extra variable that would cloud up the results, okay, the buffer is the control. So I always think of the control is a comparison group to see if there is an effect, and also it helps to eliminate other variables. All right, so to me, either one would be appropriate for part BII. On to part C. Number 1, part C, we have another additional experiment going on here. And since we're going to do part CI here that has to do with that, let's get into it. A second experiment, the scientists used plants that were identical to those in the first experiment, except that the gene encoding the DORN1 receptor was mutated. Don't forget, this DORN1 receptor is the part of the protein coming off the cell membrane that's going to accept the chemical messenger that they're testing to see if the DORN1 would initiate the cell response to close the stomata. So that's what we're testing here. Don't forget, that's the main reason that we can see if this chemical messenger, and the messengers, of course, are the AP4 dinucleotide phosphates or the CP4C. All right, those are the two things that we're testing on this DORN protein, this receptor. We're going to have to assume that this mutation on this protein receptor provides a loss of function, meaning now it's the wrong shape and it will no longer accept the chemical messenger, and it won't be able to bind to initiate the transduction signal that's going to go through the extracellular to the intracellular and eventually lead to the cell response, which is closing the stomata. So that's important that we understand what's going on here. Take a look at our study here, and let's just get right to the question for part CII. It says, justify the scientists treating one sample of leaves with the ABA in the experiments. So we have to remember that the nucleotides that we're testing are the AP4A and the CP4C. They said upstream in the previous blurb that ABA is another chemical that's not a dinucleotide phosphate or phosphatase, and it's essentially going to decrease the stomata in a different pathway, since we're not testing to see if ABA works through the DORN1 pathway or the DORN1 signaling pathway, however we want to talk about. So what is ABA? It's not something we're actually studying, but we're going to add and we're going to see if it lowers the relative size of the stomata. So this is a positive control, my friends. So we talked about the buffer in a previous explanation. This is a negative control because we're not going to have any closing. There's going to be no effect due to just having the buffer. There's no treatment group, so the stomata, which is sitting in light, which is going to activate it to be open, is going to stay open. And this is a positive control because we know this chemical, although we're not studying it directly, does close it. So this is going to do what we expect. So these are two controls. So adding the ABA is a type of control. But again, we need to justify the scientist treating that. So we need to give more information than a description and or just identify something. So how do we justify them treating? Well, the reason, and I come up with two that really work in my head. The first one that I think about is the fact that by adding ABA, we're essentially testing whether these treated nucleotide chemical messengers are going to use an alternative pathway other than the DORN receptor, right? One of the things we do with a control is to eliminate other variables. Maybe these chemicals do decrease the size of the stomata, but don't use the DORN pathway. And if that's the case, they would be similar in their effect that ABA does, possibly, when you mutate the protein. So think about that. By mutating this protein, we can see that CP4 still, has the same decrease as the ABA. So maybe CP4C is using an alternative pathway. So the ABA gives me a value to compare to see if one of the two or all the treatment groups are finding another pathway beyond the DORN1. Don't forget, we're mutating the DORN1 here. Okay, and that should really stop the signal of these chemicals. We should not expect any closure. And that's what's happening in AP4. So again, so for me, one of the answers would be that we're eliminating or testing to see if there's another pathway that these chemical messengers that we're testing are finding, like the ABA. or eliminating that pathway as well. So that's one way to think about it. The other thing that I think about too is that as a negative control, we're kind of testing to see if our experiment can actually measure correctly a decrease. Can a decrease be measured in this type of an experiment? So both reasonings, I think, are great answers to this question. So on to 2CII. Question 1CII, based on figure 2 above, describe the difference between the effects of AP4A and CP4C treatments on the size of stomata in plants with mutated DORN1 receptors. So here we go, continuing on the saga, dealing with how a receptor that triggers a transduction signaling pathway that leads to a smaller size or a closing of the stomata based upon these two nucleotide chemical messengers, right? That's the theme of the question. But here's based on figure 2. I highlighted that because people tend to forget that it's based upon the data here. So what we can see here is that we can clearly have error bars here. We've established early on that our buffer solution on is our control. It's a negative control because nothing is going to happen. The stomata are going to stay pretty much open. We have the ABA, which is our positive control, because we know that that's a chemical that outside the pathway of the DORN1 receptor, it decreases. Okay, that being all established already and understanding there are error bars here, when I look at the AP4A and compare it to the buffer or the control here, I can see that the error bars overlap. And if they overlap, there is no significant difference because remember, the standard error, the mean, really is the range of where the true mean can be. It's a reliability measure, right? It's a measure of confidence. And so the confidence values here overlap, meaning that these values are considered to be essentially the same. There is no significant difference. And that's key here. So anytime you have an error bar in your graph, you must talk about significance. Now, when we compare that to CP4C, we can see that there is going to be no overlapping of the error bars here. So the treatment group with CP4C has a significant decrease in the size of stomata. There is more closing going on here when there is a mutated DORN1 receptor. So my answer is very simple. The AP4A test group had no significant change in the relative size of stomata, while the CP4C test group had a significant decrease in the relative size of the stomata in plants with mutated DORN1 receptors. On to question 1CII. Question 1CII, activation of DORN1 receptors induces plant cells to produce certain molecules that cause the stomata to close. Now, these certain molecules that they're talking about result from the binding, or I should say the reception of the, in this case, we're talking about AP4A, that's the chemical messenger, into the receptor that's going to have a transduction signal that somehow produces either secondary messengers, or those secondary messengers activate protein kinases who do the phosphorylation cascade that leads to either the production of other molecules or activation of other molecules that eventually leads to some chemical reaction or process or mechanism that closes the stomata. That's the process here. This is a signal transduction question overall in terms of its theme. So the question is, with a mutated a shape of a receptor that is the wrong shape, we understand that we should not be able to accept the chemical messenger, and then the cell response is going to stop. So understand that we have the mutated DORN receptors that are given to us in figure 2, and for ease of viewing, I have figure 1 here, which is the first test that had the non-mutated DORN receptors. Okay, so here comes the question. Based on data in figures 1 and 2 that I have above, For cells treated with the AP4A alone, that's all we're talking about here, predict the relative production of these molecules, whether they be secondary messengers or activated kinases or the chemical itself that's produced in this chain of events that occurs in a reception transduction signaling pathway that leads to a cell response. That's this question is about. So they want you to predict, which is a nice action verb. Just tell me, what do you think is going to happen? There's no explanation here. Predict the relative production of the molecules, whatever these molecules must be, but they're downstream from the receptor, right? By the DORN1 mutated cells in comparison to the production of non-mutated cells. So let's talk about non-mutated cells first. Remember, this is all about the AP4A treatment group only. So when we look at the non-mutated cells, we can see that there's a significant decrease in the stomata size. There's a closing, right? So certainly with non-mutated DORN1 receptors, this chemical was able to close the stomata because it produced, or the reception of the AP4 to the DORN1 receptor led to the production of molecules that eventually closed this stomata. Now, when we look at the treatment group of AP4 in the mutated DORN cells, we can see there's no significant difference. In fact, there's no closing compared to the control. So, what we can say there is that there is no production or probably no reception. The chemical signal cannot bind to the mutated DORN receptor here, which does not lead or prevents the production of the secondary messengers or the kinases being activated. All of the downstream mechanism that occurs in a signaling pathway is stopped. So I would expect that there's going to be no molecules that are produced. The prediction here would that because with the mutated cells, there is no stomata closing, no significant change in the stomata openings compared to the control here because the error bars don't overlap. So there's not going to be any molecules that are going to be produced as a cell response to close the stomata. All right. So my answer to this question is for 1CII is the relative production of molecules by DORN1 mutated cells that are treated with AP4A will be less than the relative production by DORN1 with non-mutated cells. I'm explaining the answer. Of course, I don't need to explain that, of course, in my prediction. On to part D. Question 1, part DI, and I guess II because I can't really do one without the other. See, in I, I'm looking to just predict based upon a new piece of information they're giving us, but I can't predict without already having the justification. So I'm kind of combining both of these because I really can't do one without the other in its explanation. So any case, let's continue here. What is a new piece of information they're giving us? Well, they're saying the DORN1 receptor transmits a signal that results in the increase in the transcription of genes involved in plant defenses. Okay, so now we're switching gears here. We were talking about the fact that the DORN1 receptor with the appropriate ligand, in this case AP4A, that nucleotide we've been talking about, that's going to bind and to that receptor, was causing the a mechanism or transduction signaling mechanism that resulted in the closing of stomata. But now they're saying that another thing that I guess it can do is that it can produce the transcription or transcription factors. It's somehow going to allow the transcription of genes that help in plant defenses. So it's another thing that it does. The scientists then developed these DORN1 mutant plants in which the DORN1 receptor lacked most of its intracellular domain. So below the questions here, I drew a rudimentary, the best that I can, not an artist here, but the best that I can, a GPCR receptor, a gene protein coupled receptor, the most common type of receptor mechanism that we have. And we can see the receptor here is complementary to my shape. This is a model, it's not so simple. And we can see that most of the receptor is within the within the cell membrane here of the phosphobilipid layer here, the cell membrane. Now we can see the extracellular side is where the chemical signal is going to bind with the receptor. That of course has a certain specific shape that can only accept, certain chemicals that fit in that three-dimensional receptor. And when that occurs, normally what that's going to do is cause a conformational change on the intracellular side. So this alpha unit actually gets released so that it can go and activate either another enzyme or ion channel, or in any case lead to the production of secondary messengers that eventually, of course, activate kinases that undergo this kinase phosphorylation kinase cascading. That's going to activate and eventually activate other molecules and lead to, of course, in this case, the production or activation of transcription factors that are going to open up a gene and transcribe these plant defense proteins. So in this question, they're talking about the fact that they developed a mutant plant that lacked most of its intracellular domain. So that would mean that this part is missing or has the wrong shape. It's a loss of function. So even though we can still receive the chemical, because the receptors here, they're being very specific in where this mutation is. It's in a subunit that is somehow missing the intracellular side or causing a loss of function because it's got the wrong shape. So a signal still can be received by the ligand, but it's not going to be transduced into the intracellular side. Even though there might be a conformational change, there's going to be no activating of the secondary messengers or activating other enzymes that lead to a cell response. So the missing internal piece of, or lacking the intracellular domain, is vital. That piece missing causes a basic loss of function of the entire transduction signal that leads to transcription of genes. So I kind of justified my prediction by trying to understand what's happening here. So in part DI, okay, very simply, the answer to this is that the transcription of genes involved in plant defenses would be lowered or terminated in mutated cells as compared to non-mutated cells. Right? In mutated cells, you're not going to be able to transmit that signal internally that leads to secondary messengers, because we need these intracellular components of the GPCR or any receptor to go and activate other components of the cell internally. So that is the answer there for part 1D. Just very simply, the transcription of genes involved in plant would just be lowered in mutated cells as compared to the non-mutated. I like to say that as compared to the non-mutated cells, very important, because they did ask you to do what? Relative to the effect. Remember, it says right here, relative to the effect of AP4 on non-mutated cells. Okay, so that's important. All right, any case, I'm going to roll right into this, part DII. We kind of talked about the justification. All right, well, if you're going to mutate the internal side of our receptor, it can't activate other components of the transduction signaling pathway that leads to, of course, the cell response. This cell response, of course, is transcription of genes. So I wrote in my answer, the intracellular domain that is missing in the mutated receptor would prevent secondary messages from being produced, regardless if a nucleotide is bound to the DORN1 receptor. Any case, on to the next question, part 2. Question 2, part A. This is a knowledge-based question that kicks off this new theme that runs through question 2, this experimental design theme. Let's go read the blurb above the question to see what this theme is. Eukaryotic cells use small interfering RNA, siRNA molecules that regulate means control the expression of certain genes. Controlling that expression means allowing that gene to become a protein that eventually becomes a trait or not, and by binding to complementary sequences in target messenger RNAs. This binding causes the protein AGO2 to cleave target RNAs as a result of the cleavage, the messenger RNAs are not translated. So we're preventing and stopping the translation of a messenger RNA that has already been transcribed from becoming translated into a protein by these siRNA molecules that bind and then help the AGO2 cleave them. This is the overall theme of the question. Here comes a knowledge question in part A that sort of ties into this theme, although it's really just a separate question by itself. And here it goes. So describe one location where ribosomes are found in eukaryotic cells. It's very important you understand that eukaryotic cells are the more advanced cells as compared to prokaryotes that have membrane-bound organelles that make them incredibly efficient in doing the job they do, especially when we go from gene to protein. We have these membrane-bound organelles called the nucleus here, and in that nucleus we have the DNA, and messenger RNA is made and transcribed, and more important, that messenger RNA, once it is processed, we cut out the what? The introns, all right, splice together the exons, and we put the caps in that you've talked about, and then that allows it to move through a nuclear pore through a endoplasmic reticulum. And as it goes through this endoplasmic reticulum, another membrane-bound organelle, it eventually will bind with the subunits of the ribosome that's embedded in the endoplasmic reticulum. And once, of course, it binds, translation occurs and protein synthesis begins. So this is a nice little passageway, like a conveyor belt, that these membrane-bound organelles work together to go from gene to protein. So again, the question reads, describe one location where ribosomes are found in eukaryotic cells. And it's important to recognize that the action verb here is describe. Tell me something about the location. If it was identify, that action verb, you could just answer with in the rough ER. So my answer would be that the ribosomes are embedded in the plasma membrane of the rough ER. You could also say that they're also located within the matrix of the mitochondria. Don't forget the mitochondria have their own ribosomes, as they were once their own living and breathing prokaryote before they were engulfed. So in any case, on to part B, II. Number 2, part BI, using the template in the space provided for your response, construct a bar graph that represents the data in the table of this experiment. Now, what is this experiment? Really important to understand what we're doing here. This experiment, we're looking at 2 genes, and we're looking at 3 different genotypes. Positive meaning wild type, meaning you have the AGO2 gene that is working, therefore it binds to the messenger RNA when that siRNA already cleaves it, which is basically marking it for degradation. It's a part of a regulation that's happening in between transcription and translation so that translation does not occur. And so we're seeing how, and we're measuring how much messenger RNA is being transcribed with individuals with varying amounts of recessive alleles. And we're measuring 2 different genes. We're measuring the amount or the average amounts of messenger RNA that are actually available and not being cleaved. So we can see the value here. And don't forget, this plus or minus business here is standard error of the mean. It's a reliability or confidence statistic that must be graphed as well. Even though they say your graph should be approximately plotted and labeled, you need to make sure that you add the error bars. That is definitely going to be a 3rd point there. It's important because somewhere, somehow, they're going to ask you about one test group compared to another, and you need to know if there's a significant change from one test group to another. So this was kind of complicated for some because, wait a minute, we have 3 different individual genotypes, and then we have 2 different genes. But if you thought about this, a bar graph is definitely more applicable. We're going to have gene G with 3 different test groups, and then gene H with 3 different test groups. So you should recognize right away that, hey, I'm going to need 6 bars in my graph to demonstrate that. All right, so any case, let's take a look at my graph that I made here. I added a table. You didn't need one to get the points. It's just kind of something you get ingrained in doing when you write and do research papers or, science in general. Any case, so we have the relative average amount of messenger RNA in the cell. So notice on the y-axis here, I added the dependent variable. I always think it's dependent variable is easy to understand in terms of it's what we measure. We're measuring the amount of messenger RNA that's present with cells that have, either a complete working gene, half working chromosomes. When I say half working, you have one chromosome that has the wild type allele or the allele that's working, and then one chromosome that has the mutated. And of course, homozygous recessive, you have individuals with both chromosomes that are mutated. All right, so any case, so this is what my graph looked like. Notice I had the individuals down below, and then I had gene G and H alongside each other for each different genotypes. But if you notice, there's still 6 bars, and you certainly could have done this other ways. But you should definitely make sure that you have your dependent variable, the relative average amount of RNA, as your y-axis. That needs to be there. So you're definitely, I believe you're going to get a point for plotting your lines correctly with an appropriate scale. And of course, you need to label your axes. Now, part of that plot needs to have the error bars. So on to question 2bii. Question 2bii, based on the table, determine all the cells which the amount of gene G messenger RNA produced is statistically the same as the amount in the cells with the homozygous dominant genotype, or the AGO2++ genotype. So we're trying to see which of the cells produce messenger RNA that's statistically the same as the homozygous dominant, right? So statistically the same means that the error bars, right, are going to overlap. Now notice they said based on the table. That's very interesting. And I didn't understand why they were doing this initially. Now I see why, because we just made a table in part BII. But if you made your graph incorrectly, you incorrectly plotted the points, or you forgot to add the error bars, then if they said based on your graph, then they'd have to look at your graph and it'd be harder to grade. So they wanted you to make sure you were based on the table. So no matter what you did in your graph, they could just mark this wrong. Okay, so you have to look at your error bars. So we're going to see which we're going to see based on just table gene G alone, who is statistically the same between the AGO2++. So let's evaluate these values to see if they're statistically the same by looking at the error bars by the table only. So if we take a look, we have 1.0 plus or minus 1. That means we have an upper limit of 1.1 that could be the true mean of that population. And if you take the messenger RNA and AGO2 plus minus, the heterozygotes, then that's 0.9 plus 0.04. The upper limit is going to be, if you add them together, going to be 1.3, and also range through what? Okay, minus 0.04. Don't forget the minus, so that's going to be 0.5. And that's clearly going to include the 1.1 that we have. That'll be in here. So we know certainly the heterozygote is statistically the same because the error bars will overlap. Now if we look at the homozygous recessive individuals, it's 1.5 to 0.5. So the upper limit is 1.5 and the lower limit, okay, is 0.5. That's clearly going to overlap the 1.1 as well. So the 1.1 will be overlapped by the AGO2 heterozygote and will be overlapped by the homozygous recessive individuals. So both of these groups, okay, are statistically the same as the homozygous dominant because of the error bars overlapping if you use the table. Now, if you made a nice accurate graph, it's even easier to see. So for gene G, you can see that the error bars here, the error bars here, and the error bars here are all going to overlap. And clearly, heterozygotes and the homozygous recessive individuals are statistically the same for the amount of RNA as compared to the homozygous dominant. On to question part CI. Question 2, part CI, based on the table again. They don't want you to base it on your graph because your graph could be wrong. They don't want to do double jeopardy here, so they want to just be able to get the right answer. Describe the relationship between the number of wild type copies. Wild type copies, that should ring a bell. Those are the plus values. And again, if you didn't know, they give you that up here. The wild type are the homozygous dominant. So how many wild type copies? You notice we have a homozygous dominant individual, we have a heterozygote individuals, and we have homozygous recessive individuals, right? And as a little reminder here, each positive represents a dominant allele or gene that's not broken, not mutated. Remember, they come in pairs, right? So if you look at the homozygous dominant, there's two genes that you carried on two different chromosomes, homologous chromosomes, that are working genes. And if you notice, they said there is a non-functional gene, and that would be in the recessive allele. So that means that one copy of the heterozygotes, plus negative, is recessive and not working. And of course, two copies are not working in a homozygous recessive individual here. So when they talk about the number of copies of wild type alleles or genes, they're talking about these pluses here. How many individuals have a working gene that's positive? So back to the question, based on the table, describe the relationship between the number of wild type copies, that means how many chromosomes are in the population that are carrying the positive gene, or the gene that's dominant and working, of the AGO2 gene. That's of course what all of this is about, and the amount of gene H. So this is where I think a part of the question 2 that people started to have trouble with. From part C on, it got a little bit challenging. And I don't think this is a challenging question, especially if you got yourself a good graph. Hard to really see this on the table. I will try for the table and then go to the graph. So again, we're looking to see a relationship between, and when they say describe, tell me if it's as one goes up, the other one goes down. Those kind of descriptions are important, and that's the type of description you're going to need here. So if we look at the graph and we're looking for gene H, all right, and we're looking here and we're trying to see the relationship. Well, we should know right away as we move to the left, right, to the Beyonce, we can see the number of alleles or wild type copies are increasing. We have no wild type copies here. We have 1 out of 2, and then we have 2 out of 2, right? There are more wild type copies as we go from the homozygous recessive to the heterozygote to the homozygous dominant. So you should see that we move to the left. When we move to the left, those copies are increasing. Now, what's happening in gene H? Be careful you read this carefully. It's easy not to. Okay, in gene H, what's happening? Well, you can see, party people, that the amount of messenger RNA that is being produced is decreasing as the amount of wild type copies is increasing. If you consider the error bar range of 3.0 plus or minus 4, hey, this is going to be 3.4 to 2.6, and that's still outside the range of 2.1, right? This would be plus or minus 2.1, so 2.1 to 1.9. So clearly those error bars are going to not overlap, and there is a significant difference. And of course, if you look at the 2.1 to 1.9 range, that is not going to overlap a 1.0, or in this case 1.1 to a 0.9 range. So just doing the quick math of the error bars, you can clearly see as we go to the left, the wild type copies are increasing. There are more chromosomes that are carrying the dominant allele. We can clearly see that there is a significant decrease in the amount of messenger RNA. So as the wild type increases, the amount of messenger RNA produced is decreasing. I think it's easier if you have an accurate graph, though, to read that. They didn't want you to do that because you might have an incorrect graph, and they did not want to grade this based upon double jeopardy. So if you look carefully, and we have here, we can see that as we go to the left, the amount of H is clearly significantly decreasing. Okay, so hopefully that makes sense and you describe that accurately. On to question CII. Question 2, part CII. Let's go get it. Based on the data in the table, they don't want you looking at your graph that you made because it may be wrong. So they want you to look at this information here to get the right answer. For gene H, this is when I first read this question, I did not see gene H, so you have to read carefully. So we're looking at gene H here, and we want to calculate, again, there's my action verb, calculate the percent by which the average amount of the messenger RNA cells increases from AGO minus, that's the homozygous recessive, how it increases compared to the original or the initial AGO2 plus minus or heterozygous test group. So we're after the percent change here between the increase that occurs from the homozygous recessive individual test groups to the heterozygous test groups. As we can see from the data, looking at gene H, when I have the heterozygotes test group, and I can see from the data table with the heterozygote test group that for gene H, the amount or the average amount of messenger RNA that was produced was 2.0. while the average amount of the homozygous recessive was 3.0. So they're asking again, go back to the question, keep rereading, calculate the percent by which the average amount of messenger RNA in the homozygous recessive increases compared with that to the heterozygote. There definitely is an increase from 3 to 2. They want you to calculate the percent change. They might not come out and say the percent change, but they want the percent difference between the 3.0 increase from the 2.0, right? So this, what happened here from 2.0 to 3.0 between these two test groups for gene H? What was that percent change? What was that percent increase? It means the same thing. So I've always found it weird that they don't give you the percent change formula. I was told by someone from the College Board, students should know some basic statistical operations like sum, addition, divide, or multiply. I get that. They actually give you a formula for the average, yet they don't give you the formula for percent change. Kind of odd for me. But in any case, percent change is take the final, which is the larger. It could be smaller, that it could be a negative percent change. But in this case, take the final minus the initial and divide by the initial. And then of course we're going to times that by 100. And when you do so, you get a 50% change, or 50% change from the heterozygote group to the homozygous recessive group in measured or average relative RNA. Okay, so that's the calculation. You needed a percent change formula. Hopefully you did that. We did that in a couple different labs, and that's what they were after. So on to question 2, part D. Question 2, part DI. The scientists claim that the siRNA, the small interference RNA binding, and the AGO2 cleavage of messenger RNA plays a greater role in gene H expression than in gene G expression. Based on the table, they don't want to base it on your graph because your graph might be wrong. Support the scientist's claim. So we're not evaluating, we're just going to support this idea from the data table. Okay, and again, this is important. The theme of this question was that we have small interference RNA, right? And RNA is single-stranded. Are there times where it binds to itself? Sure, tRNA. But the single-stranded RNA, okay, is going to find a complementary strand somewhere on another messenger RNA that was transcribed that is headed for translation. And this binds. When this binds, it creates some sort of marker for the AG2 to cut up and cleave this messenger RNA so that it cannot be translated. This is one of the gene regulation mechanisms that eukaryotes have. Now, what we're looking for is how can we support the claim that the gene H expression is affected more by this mechanism of gene regulation by messenger RNA, more so than gene G. That's what they're saying. They're saying gene H is able to be regulated by this mechanism of binding to messenger RNA that's targeted for translation, and then AGO2 now binds and cleaves that messenger RNA. So how can I look at that? Well, if you look at the table, they're telling you to look at the table. I'm telling you it's easy to look at your graph if it's accurate. But I'm going to go to the table. How can we see that gene H, okay, is more affected by that pathway? Well, what we can see is, and this is important, if you look at the individuals or the type of individuals in each test group, homozygous dominant, when we look at gene H, we can see that there's a 1.0 plus or minus 0.1 value for the amount of RNA. And as we move across to the different test groups, we have a heterozygous, okay, test group and a homozygous recessive. As you can see, the amount of pluses decrease, or the amount of working genes are decreasing, and what's happening is the relative amount of messenger R is increasing significantly. You can check the error bars, 10.01. If you add that, this is going to be 2.01, 3.04. So you can see that these ranges, considering the plus or minus standard error of the mean values, make this gene H to be significantly increasing in the amount of messenger RNA that's available. So we can see there's an increase in the amount of messenger RNA that are not targeted for regulation and cleavage by the AGO2. So as you have an increase without the working gene that produces the AGO2, and so this type of data supports the scientist's claim that gene H must use this type of gene regulation because there is significantly more uncleaved messenger RNA when there is less functioning genes. Or better yet, there is a significant decrease in the amount of messenger RNA available that's being regulated by the AGO2 cleaving it when we have a greater number of working AGO2 genes if we move toward the left of the table. But what about gene G? Well, as we can see in gene G, same test, as we move across this 3 test groups, the amount of alleles or chromosomes that have the working gene is decreasing, but the amount of messenger RNA is statistically staying constant, because if you check out the error bars or the standard error of the mean, these will overlap. There is no significant difference between these values. As you decrease the ability of the individuals to cleave messenger RNA, their ability stayed the same. Where in gene H, when you decrease the ability of the individuals by having less working alleles or genes, it increased. So clearly gene H uses the AGO2 mechanism to regulate messenger RNA in gene H more than gene G. As an aside, if you look at the graph, if you've got yourself an accurate graph, it's so much easier to answer these questions. So if you take a look at gene H, you can see using the error bars that there is a significant increase in messenger RNA that's available as the number of working genes decreases. And if you look at gene G using the error bars again, there is no statistical difference here because the error bars all overlap. So there's no change when we decrease the number of working genes, which means, of course, that gene G is not using that regulation pathway and gene H is. On to part DII. Question 2, part DII. Anaphase 1 of meiosis is blocked in mice with AGO2 minus genotype. That's the homozygous recessive genotype. That's the genotype where we do not have any functionally working AGO2 genes. Scientists hypothesize that block in meiosis occurs because of the continued presence of a spindle fiber stabilizing protein that prevents the chromosomes from separating. Based on the table, of course, they don't want to base on your graph because, again, your graph might be plotted incorrectly. Explain how, that's important, explain how the homozygous genotype could cause this effect on the dividing cell. So that we have the AGO2 gene. If it's working, you're producing this protein that is going to cleave. Remember, the whole purpose of this question or the theme is that it's going to cleave messenger RNA, and when it does so and cleaves it into pieces, it no longer can translate. So translation is impaired. Remember, this is a regulation. This is how we stop messenger RNA. This is how we prevent expression of a certain gene at the messenger RNA level. So that's what AGO2 does. But here's the problem. Because we have a homozygous recessive, we make a gene that can't do this. It can't cleave messenger RNA, which means it can't stop translation, which means translation is going to occur. There's no break here. So there's nothing to stop the messenger RNA. If we have AGO2, there's nothing to stop this messenger RNA that in this case is coding for the production of this spindle fiber stabilizing protein that prevents chromosomes from separating. That's the answer. The answer is very clear. A mutated AGO2 gene, and that's all you have with homozygous recessive individuals, cannot cleave the messenger RNA that's coded for a certain gene that is going to keep chromosomes from separating. So therefore that gene or that messenger RNA gets translated into, guess what? More spindle fiber stabilizing proteins that keep the what? Chromosomes together and block meiosis. Okay, but we have to be careful how we answer here. Always reread the question. So everything I said is correct, but remember in part DII they said based on the table. Explain how AGO2 homozygous recessive individuals could cause this effect. So we need to talk about the data somewhat. So if I look at the table or I look at the graph, I'm going to look at my graph because it's clearer, because my graph is accurate, but either way it's going to work for you. Homozygous recessive individuals, guess what? Have the greatest amount of messenger RNA that's not cleaved. That's showing that those individuals do not have a working gene that can regulate and cleave the messenger RNA. So it's important that you make that part of your answer. So part of the answer, how I'm going to say, is that AGO2 minus individuals have the largest relative amount of messenger RNA for the gene that's regulated by the siRNA and AGO2 due to both chromosomes carrying a non-functional gene. In the case of mice, homozygous individuals cannot cleave the messenger RNA for the spindle fiber stabilizing protein because their AGO2 genes have the incorrect form and function. This results in the spindle fiber stabilizing protein to be continually translated and to continually block meiosis. So remember, I had my answer. I was working on the second part, but I needed to point my answer in the direction of the data. All right, so that's important. So on to now question 3. Question 3. This question is about the exposure of cyanide to cells, mammalian cells specifically, that can undergo an aerobic pathway, a pathway without oxygen that is part of what we call the fermentation pathway, or with oxygen available can go the oxidative phosphorylation pathway, which of course requires a mitochondria. So the hook here is that cyanide is a chemical that affects the enzyme cytochrome oxidase. They're calling it CCO. And they're telling you specifically it's a key enzyme in the mitochondrial electron transport chain. That should ring off some bells. Hey, that's part of oxidative phosphorylation. That's the aerobic cell respiration that we have studied. So they're saying that high concentrations, the CCO activity is much lower, and that low concentrations, the CCO activity is a little bit higher. Okay, and then they studied that with some treatment groups, and we're going to go on to part A now to see exactly what they're looking for. But again, it's important to understand what the theme is, and already just by reading quickly, I can see that this could be about stopping the aerobic pathway, cell respiration, and hooking into the anaerobic respiration. So here comes the first question, and we're seeing in part A, just like we're seeing in the first questions of many other groups of questions, a knowledge-based question. So here we go. Describe the advantage of cells producing ATP from one molecule of glucose by aerobic respiration rather than by glycolysis alone. So it's important we understand that we're talking about aerobic respiration. This, of course, is going to be the idea that we're going to break down glucose. So we're going to use glycolysis. We're going to make pyruvate, and that pyruvate is going to move into the mitochondria and get its way into the matrix, where it's going to be oxidized further and get into the Krebs cycle, and then of course get chopped up as CO2, and in the process, in the Krebs cycle and in parts of glycolysis, we're going to produce NADH, and we're going to produce FADH2. These are the electron carriers that when we oxidize glucose, pyruvate, or any of the glucose derivatives, these are holding on to high energy electrons. And these high energy electrons are what feed the electron transport chain on the inner membrane of the mitochondria, and of course causes the pumping of protons into the inner membrane space using the proton motor force, of course creates ATP. So make sure we understand that. So we're comparing aerobic respiration with just glycolysis alone. And don't forget that glycolysis alone doesn't actually occur. It's really fermentation. But in any case, this is a knowledge-based question. Describe the advantage to cells producing ATP from one molecule of glucose by, hey, the complete aerobic respiration. That's glycolysis, that's the oxidation of pyruvate, that's the Krebs cycle, and then of course the electron transport chain to make the greatest amount of ATP. And we should be aware that very quickly that the aerobic respiration is a reason why cells use that, because there's tremendous amount of energy given off. So aerobic cellular respiration produces about 34 ATP, while glycolysis nets only 2. So that's really the answer there, and that's just something you needed to know. I don't think you needed to have the numbers, although that's pretty impressive. So yeah, aerobic respiration produces more ATP than glycolysis per one molecule of glucose. And again, one molecule of glucose produces about 34 ATP with cell respiration and about only 2 ATP. So there's a greater amount of ATP produced per molecule of glucose. All right, and that's really what they're after there. So this is a knowledge-based question, as we're seeing for most first questions in the FRQ section. On to part B. Number 3, part B, identify. Everyone loves the identify action verb, so we just got to find it. So they want you to find the 2 treatment groups that served as controls in the researcher's experiment. All right, so there's a quick way here and there's a more difficult way. So if we're just going to jump into the study, let me just go what I think is the more difficult way. Sometimes jumping into what they're saying could be difficult, time consuming, and a little bit of a mental, I would say, gymnastics here to understand what's going on. But let me just try to read this and make some sense to this for you if you had some trouble. And it will help you understand this little blurb in between before table 2 here. Some strains of bacteria produce cyanide, which then they release into their environment. That's key, okay, that they release into their environment. We're going to utilize that in a second. Other strains of bacteria do not produce cyanide. All right. And researchers grew both types of bacteria, the ones that produce cyanide, the ones that don't, in high concentration nutrient medium, and then isolated the medium from the bacteria. This is where it gets, you've taken a multiple choice part of the test. I can get being tired and saying, what are they talking about here? Well, we're talking about the fact that if you're a cyanide producing bacteria, you release that into your environment, which is going to be, guess what? That's going to be the media which you grow in, or the medium which you grow in. So that part is one thing. Now we're going to basically separate the bacteria that doesn't produce cyanide and the bacteria that does from their growth medium, right? We're going to separate that. And then they say here 1/2 of each medium sample was left at high concentration, while the other concentration half was lower. This is where it gets a little bit confusing. So let me try to explain through the petri dishes here that I have to the left. So the orange petri dish, okay, is the cyanide producing bacteria. The little black specks represent the bacterial colonies. And the blue petri dish here represents the bacteria that do not produce cyanide. Now, because they said in the little blurb above here, some strains of bacteria produce cyanide, which they release into their environment, we can bet that the cyanide producing bacteria here is releasing cyanide into the growth medium as well. Now, they said that they grew both bacteria under high concentration nutrient medium. They got a lot of glucose, they got a lot of nutrients, they're going to grow vigorously. Now, here's the part where it gets confusing. All right, now we're going to isolate the medium from the bacteria. So we're going to separate the bacteria. We're going to remove the bacteria. Right? We're going to remove the bacteria. And what you're going to be left with is a petri dish that still has the cyanide in it, because we said that cyanide's released into the environment. And then we have the other dish, can't reproduce that circle very well, is going to have no cyanide. So we remove the bacteria. And now this is the part. We're going to split these two, because the next part they said is that 1/2 of each medium was then left at high concentration. So this half of the petri dish was left, okay, as very high concentration, and then this one was cut in half somehow. So maybe they took this and into a petri dish, they separated it and spread it out. Okay, same thing here. They kept one at high concentration, and then what did they do? They probably broke it up and separated and diluted this into another petri dish. All right, so they kept some of the medium that the bacteria originally growing in, okay, were under high concentration of nutrients, and then they diluted, okay, they diluted the other. Okay, so basically they have 4 samples, right? And that's the part where people now should get this. Now we have concentration is high, all right, and that would be these values here and here. And that medium that they grew cells that are mammal cells, right, into that medium was high. And there's going to be a place where there's high cyanide. That's going to be where this medium was not diluted. It was grown with bacteria that produce cyanide, so there's going to be a lot of cyanide there. And then we had another petri dish where there was no cyanide, that would have this half of the medium, all right, that we put mammal cells to. Then we're going to have a concentration that has low, that's the one we diluted. I'd probably prefer they said that. A lot of medium words in here. But then, okay, we have the diluted version here. Okay, that's going to have some cyanide, but not as much, because we diluted it to lower the concentration, lower the amount of cyanide that was released into the environment. The medium's the environment. And then of course we have the low concentration with no cyanide, and that is this right here. Okay, so those are our 4 test groups. So that, to me, a little more complicated to understand, but I really think it's important that, to think through this, because this is what the AP wants you to do, be able to understand their setups. Now, could you have done this without all of my diagrams and understanding? You probably could have, because I said there was a fast and slow method of doing this. I did the slow method, or at least the method that takes longer to explain, but I think longer to answer. So we go back to the question, part B, identify 2 treatment groups that serve as controls. What I'm going to do here is look at the name of the study. Experimental design to test effects of cyanide on mammalian cells. Okay, I'm looking to test the effects of cyanide on mammalian cells. So my test groups that I'm trying to test are going to have different levels of cyanide. Here you go. So these are my 2 test groups. I'm not really testing the effect of no cyanide. I want to see what cyanide does, either with high concentration, too low. And what I'm drawing arrows to is we need to compare how that would occur if there was no cyanide. So if you have high concentration medium, which is, by the way, if there's cyanide, we want to compare that to high concentration medium with no cyanide to see if there is a difference, right? That's the reason for a control. Or we want to eliminate the medium as a factor here. However you want to think about using control. Remember, controls are used for 2 reasons. So clearly my controls are 3 and 4 here. Why? Because they're the groups that don't have cyanide that we need to compare. And because the experiment was based upon to test the effects of cyanide, I need to what? Compare it to test groups that don't, so that I can see if there is actually an effect. So that's why 3 and 4 is it. And again, just identify. I go into a lot of explanation here because I want you to understand the problem. Understanding more of these problems gets you ready for more types of problems like this. That's the thinking the AP or the College Board wants you to have. So letter B is 3 and 4. On to part C. Question 3, part C, based on the information provided in both tables, including this table right here and the table above, we're going to do what here? We're going to predict the treatment group that is likely to have the highest ATP production in all treatment groups. So if I want to think about highest ATP production, I'm thinking from question part A, I'm thinking who's going to have the greatest amount of cellular respiration. So the one thing I have to recognize is from the blurb, if you haven't read this part, went to your questions, you need to understand that they said early on cyanide's a chemical that affects the enzyme cytochrome C oxidase. It's a key enzyme in the mitochondrial electron transport chain. So if there's cyanide present, it's going to decrease the enzyme activity in the electron transport chain, which is going to minimize the amount of ATP made in the mitochondria, which is through aerobic respiration or oxidative, specifically oxidative phosphorylation. So it's important to recognize that when we're trying to figure out who's going to produce the greatest amount of CO2, we're looking to see which one is going to have the greatest amount of this CCO activity, or the enzyme that contributes to the mitochondria successfully completing cell respiration. There is more energy in cell respiration. They set you off with that idea in the first question. Now, when you look at the effects of cyanide on the activity of that enzyme that pretty much is going to run or not run cell respiration in the mitochondria where there is more ATP, we see when there's no cyanide, there's going to be normal activity, which we would expect the amount of ATP to be normal for aerobic respiration or cell respiration. When there is low cyanide, look at this, there's a slightly increased CCO activity, which means higher than normal. That's the key here. There is higher than normal. Okay, that's really important. Where in high cyanide, as the blurb suggests, there's going to greatly decrease CCO activity, which means there's going to be greatly decreased, cell respiration, specifically the part where produces the greatest amount of ATP, which of course is oxidative phosphorylation. All right, not sure why I'm writing letters there. All right, so that's important that we have normal amount of ATP, but then we have slightly increased and of course greatly decreased, what they describe. So we're looking for a choice or a test group that's going to have just a little bit of cyanide, and that should give me a little bit greater than ATP production. We're looking for a test group that's going to have a small amount of cyanide, which is going to slightly increase the CCO activity, which is going to increase the ATP production even a little bit greater than normal. And again, if you're lost in the sauce, reread your question. Predict the treatment group that's likely to have the highest ATP production of the four groups, of the four treatment groups. So I'm going to look for a choice that has a low amount of cyanide. Now, if you're with me with part B, I left that information up here where they went through this blurb here to explain the treatment groups. We understand that cyanide producing cells are put in a medium that had high nutrients, and non-cyanide producing cells in the blue here were put under also high nutrient medium. Then, as we talked about, we cut these petri dishes in half. We removed and separated the bacteria from the medium, and then we cut these medium in half, allowing one side to have high amounts of concentration and the other is diluted, right? The other ones were diluted, and that gave me 4 test groups. Now with that medium with high concentration that was grown with what? Cyanide producing bacteria, that is going to be right here. And then the diluted that was grown in cyanide producing bacteria is right here. That's that treatment group. But there's not going to be as much cyanide because it was diluted, right? And then of course we put mammalian cells in there, and we I guess tested the ATP production. They don't give us that data, so we have to predict what the outcome we think is based upon this idea right here. Low cyanide slightly increases the activity of an enzyme that's responsible for producing ATP and the largest ATP production part of cell respiration, which is oxidative phosphorylation. So clearly the answer is going to be choice 2. That's the prediction we're going to make. Now, reason why it's a prediction, they haven't done the study, right? So we're going to predict that that's going to have the greatest amount of ATP, because that's the one where it's going to have a not only an enzyme that's working a little better than normal in the mitochondria where most of the ATP is made. All right, so again, part B identified 2 treatment groups. So in part C, predict the treatment group that's most likely to have the highest ATP production. All you have to say was treatment group number 2. On to part D. Question 3, part D. Researchers claim that cells in which CCO activity is inhibited, key to the problem right here, will show an increase in lactic acid production. Support the researchers' claim. Oh, you should love this action verb. We're going to support the researchers' claim. All we need to do is explain it, right? We don't have to figure out if it's right, wrong, not changing, increasing, decreasing. So this should be a welcoming action verb. Any case, the claim is that the CCO enzyme activity is inhibited. Will show an increase in lactic acid. Now, what does that mean? Well, we know from the previous information that's posted in this question that a decrease in CCO activity is going to lead to a decrease in ATP produced in the mitochondria, or a decrease in aerobic respiration, okay? Because we're essentially going to inhibit the electron transport chain. Okay, they told us that this is a key enzyme in the electron transport chain, which means electrons will not be transported from complex to complex in the inner membrane of the mitochondria, which means protons will not be pumped into the intermembrane space. They won't run through the ATP synthase, and we're not going to make the majority of the ATP that can be made by oxidative phosphorylation, by the aerobic pathway. So that's important. So if we're going to shut down the aerobic pathway, then the anaerobic pathway to make energy must occur. So in my diagram below here, I'm showing that a decrease in CCO activity is going to lead to a decrease in aerobic respiration in the mitochondria. And this is going to prevent electrons who are being carried by NADH, who originally got those electrons by breaking down glucose either in glycolysis in the cytoplasm or breaking down glucose in the Krebs cycle and delivering them into the inner membrane of the mitochondria for the electron transport chain. Well, if we're going to inhibit the electron transport chain, well, that means that the NADH is not going to be used here, and more importantly, we're not going to recycle it back into NADH plus, which we need to keep the glycolysis going to make energy, even if we're in the anaerobic part. Understand, party people, we're shutting down this part. NAD plus is not going to be made. We're going to back up the NADH. So this is why we need fermentation here. Fermentation is going to essentially recycle the NADH that's backing up, that's not being used, let's say, in the mitochondria, and it's going to be recycled. And of course, it's going to deliver its electrons and reduce pyruvate, that's the end product of glycolysis, into lactic acid. So the fermentation pathway is going to kick in here when we shut down the aerobic pathways or the mitochondria, or specifically oxidative phosphorylation. And you need to know, in my opinion, that when fermentation is involved, the reason for fermentation, of course, is to continually make the ATP that's needed for energy. We're not going to make as much, but we still make some to keep this cell alive. Energy is needed. You know, cells require a constant supply of energy. They're non-spontaneous, but the most important part is that we got to keep recycling the NADH it's made when we break glucose down into back Na+. That's why the pyruvate that's made, instead of going to the mitochondria, and it won't, as if there was no oxygen, it gets converted into lactic acid, which is the byproduct of recycling the NADH back into the NAD+, so we can keep this process alive. Keep this process alive. All right, so what I said was cells in which the CCO activity is inhibited will demonstrate an increase in lactic acid production due to decreased activity of the electron transport chain. If oxidative phosphorylation is compromised, then the cell will utilize the fermentation pathway to recycle the NAD+ by reducing pyruvate into lactic acid. In my opinion, you needed to talk about how the fermentation, anytime fermentation is part of any answer, you should talk about the recycling of the NADH back into NAD+. That will only occur if pyruvate, okay, is reduced into lactic acid. Now, I don't think you need to. Probably the College Board would accept something less specific, but I would definitely encourage my students to make sure they always talk about that. On to question 4, part A. Question number 4, part A, describe. There is our action verb here. One characteristic of a chromosome movement during meiosis 1. All right, so this question, its theme is about meiosis. And if you look at the little blurb they gave us here, the process of meiosis enables animals that reproduce sexually to produce gametes. And of course, gametes, you should be aware, has half the diploid number, right? We have a 2n number, which is the diploid number, and then it's going to go, when we make gametes, to the n number, half that, and that's the haploid number. So that's the theme of this question. But back to the question, describe a characteristic of meiosis. Well, so in my description here, we're going to have the total number of chromosomes into daughter cells, so those daughter cells or those gametes can now combine with other gametes and make a zygote with a complete 2n number. So it's important that we have. Now, of course, that halving occurs twice, because when we show up to meiosis, or I should say prophase of meiosis, we're going to have double the amount of DNA, because in the S phase of the cell cycle, DNA is replicated. Okay, all of that is great, but what is the movement during meiosis 1? Well, they kind of gave this question away with their diagram. So if we look in part C of the diagram on the same page, they give you the answer, right? They're showing separation of homologous chromosomes. Now, yes, they're showing an example of a problem that's called non-disjunction occurring on one side, but the correct separation is right here. So I would think this is a low-hanging fruit problem that you should have gotten. So they're kind of showing you that. Now, of course, understanding that meiosis you need to have twice. So let's just pretend we have an organism that shows up in prophase like this, and of meiosis, and we call this a tetrad. And as I said before, we're going to make this the maternal set of chromosomes, and this the paternal, and they're duplicated because of the S phase again, and they're going to line up on the metaphyseal plate, and they're going to separate into two daughter cells. That's the first halving. Remember, we have to have a 2n number that becomes an n number, diploid to haploid. We have to have our gametes to have half. So we have to separate the homologous chromosomes first, and that's really the answer here. The second halving would have to what? Have the sister chromatins. Really the only way. And that's the only way we can make what? 4 daughter cells from this initial set. Now, of course, this would be a 2n equal 2 individual. You say, how is it 2? Remember, these are copies, right? So you just count one of each, right? So there's 2 chromosomes here. They're showing up duplicated, so we can have twice to essentially get to the n number. And you can see that at the second halving, we have an n number equals 1, which is half of what we have here, 2. How do you do that? Well, you have to separate the homologs. And if you didn't know that these were the homologous chromosomes, understand that the dark copy, the paternal, is the same type of chromosome as the light one from the mother. Just carries possibly different types of the same type of genes. Could be the dominant allele for a certain gene, and the mother could be the recessive in the same spot. That's what homologous chromosomes. They carry the same group of genes, just different forms possibly. Could be the same, but different forms, and that's what diversity comes from. So we have to separate these homologs first, and that's what we do in meiosis 1. And then meiosis 2, we're going to separate the sister chromatins, and that's what these guys are here. These guys, of course, are exact copies of each other, and they're connected by a centromere. And eventually, you're going to get, okay, individual chromosomes of half of the original after the second halving. So you needed to know for this question, and I guess you could consider it to be a essential knowledge question, that we separate the homologous chromosomes first. All right, so again, the answer very simply for 4A, homologous chromosomes will segregate, and I said will segregate to opposite poles during anaphase of meiosis. So I was kind of specific, and again, if you're not sure how to answer the question, keep rereading the question. Describe. So tell me something about one characteristic of chromosome movement during meiosis 1. You definitely could have said that the homologous chromosomes, the tetrad, align themselves in the metaphyseal plate in metaphase. You could say, and I just said that they separated during anaphase. So whatever movement, they move to the center. Remember, they move as a group, a tetrad, or 2 homologous chromosomes connected by cohesion proteins. And then you could also say they're going to move to opposite poles during anaphase, where we're going to separate the homolog. So a couple of different ways to answer this. Okay, I always kind of go with the anaphase. I know the movement's so important. They move to opposite ends to create two different daughter cells. Remember, there's two daughter cells for meiosis 1, and of course there'll be 4 after meiosis 2. All right, so a couple different ways I believe to answer this. The way that I'm going to describe, or I'm going to talk about the anaphase, because that to me is the most important. And I've seen this in other practice problems where they want you to know that the anaphase separates to opposite poles. And you needed to know that it was the homologs that are either moving to the metaphyseal plate or pulling apart and breaking that cohesive proteins to opposite poles. Again, I think there's a room to wiggle here in terms of which way you describe that, but definitely homologous chromosomes should be the forefront of what you're describing. On to part B. Number 4, part B, explain. That's our action verb here. All right, we need to explain, we need to define how something works or give reasons or justifications for something. And that something is why chromosomes are more visible when viewed under a microscope during mitosis or meiosis than any other time in the cell cycle. Well, if you're in mitosis, in meiosis, you're either in that what we call the PMET, right? Prophase, metaphase, anaphase, or telophase. If you're not in these parts of the mitosis or meiosis, we say we're in interphase. An interphase, my friends, is a part of the cell life cycle, whether you're in G1, S1, or G2, right? We're not ready to actually separate DNA into daughter cells, whether that be in mitosis or meiosis. And to separate DNA, we're making a complete copy in mitosis, or we're having twice in meiosis. To separate DNA into daughter cells, you can't do it when it's in the open, unraveled format. So we're going to need to essentially coil it up. Okay, make this chromosome scaffolding of what we call chromatin. We need to coil this up so that it becomes one structure that can be used. See, when DNA is being used by the cell, of course, the cell doesn't use all the DNA, it uses just part, but it needs to be open enough so RNA polymerase can come in and make a copy, and we can go the gene to protein method here. So if it's always coiled up and closed up, and that's one type of regulation we have in eukaryotes, then it's not open for any usage. And of course, we need our DNA, of course, to keep making proteins to interact with our environment and so forth. So in interphase, the DNA is more open. In a eukaryote, it's still wrapped around these histones, because actually the amount of DNA that we have in human cells is actually very, very long. They're linear chromosomes, and I'm going to draw these histones here. And you should be aware there's these proteins that DNA is wrapped around, and that's called chromatin. And again, these are just some important knowledge base ideas. And we can open and close these up where we need to for cell regulation at the DNA level. But what this is all about, this question, is why chromosomes are more visible when viewed under the microscope. Well, one of the ways that we identify that we're getting into prophase, whether that's meiosis or mitosis, is this chromatin is actually going to coil up and scaffold into a more wound up structure called a chromosome, right? So these things are going to make like big loops and keep going, and those loops are going to create this very dense DNA structure called the chromosome. And so the answer is that when we're in prophase, one of the ways to identify prophase is to see that we can visibly start seeing chromosomes that are now wound up, or the chromatin wound up into chromosomes. The chromosomes are visible under the light microscope, and the word that we use is that these chromosomes condense, right? That's where the extreme winding up. And the greatest example I can give you, if you want to separate spaghetti on a plate evenly, and it's just thrown on the plate haphazardly open, that's going to be very difficult. You're going to break strands. And if that's DNA and you're going to break strands to separate them, you're not going to separate them evenly, which is going to mean that your offspring and have the wrong number of chromosomes and all kinds of other problems, but they also will break. So what if every chromosome or every piece of spaghetti wound up very tightly, and then now they're going to be easy to move around to give the hereditary patterns that we need. So yeah, the best way to explain this is that in prophase, these chromatins condense. Do you have to say chromatin? I don't think so. My answer was during prophase, chromatin, loosely wrapped DNA around histones, I added that because I guess I'm showing off, is coiled up or condensed into a dense structure called a chromosome. That's what they're looking for. All right, that word condenses something, the idea that in prophase. And by the way, if you did the very root tip onion cell lab, or whether you looked at slides or did that, you knew you were in interphase when you couldn't see the what? You couldn't see the cells. Then maybe you saw it, this is a bad drawing, but you just saw some, dark areas. But when you started seeing some distinct clean areas of chromosomes, you knew you were in prophase. And that's one of the ways that we recognize that we're in prophase, is that the chromosomes condense. All right, so again, on to number 4, part C. Question 4, part C, the little blurb they give us here. So in the middle of these FRQs, they do change up the questions, and they may change up the conditions. So it's really important that we definitely get into this. An animal cell with 2 pairs of homologous chromosomes, a diploid number of 4, has undergone meiosis to produce 4 gametes down here. An error occurred at one step. Hey, that's right there. That's that non-disjunction as shown in the figure. One of the sets of chromosomes is represented in white and the other set is represented in gray. And these sets, of course, are homologous chromosomes. Okay, so yeah, we have a 2n equals 4 here. We should recognize that we have a total of 4 chromosomes. There's 1, here's 2, here's 3, and there's 4. Now they are duplicated and connected by a centromere, and of course these individual ones are sister chromatins, and of course the different colors of the same type are homolog. So this is chromosome 1 that's bigger, these are chromosomes 2. Okay, we definitely have non-disjunction, and we have a different number of chromosomes that are produced in the daughter cells. Because of non-disjunction, these are the N+1 cells. These have 1 extra. N means the haploid number. If we have a 2N equals 4, okay, because we have 2 different sets of chromosome pairs, one from the mother, one from the father, that's 4. And under 2 halvings of meiosis, we should have an N number that equals 2. Well, if you notice, we have 3 here and 3 here, so we're N+1. So these are N+1 gametes. And the N number needs to be 2 here as well on the right-hand side, so these are N-1, so 2-1 cells. Right, because these have an N number of 1 because of the non-separation of the homologous pairs during for chromosome 1. That's the error there. Okay, so we've got this set up. So now let's continue on with their continuation, or let's continue with the blurb and finally the question. Assume that each gamete produced by the cells shown in the figure is fertilized by a gamete with a complete set of haploid set of chromosomes. That's important to know. Okay, so we're going to fertilize these gametes. Every one of these are going to be fertilized. Okay, and they're going to be fertilized with another individual that is 2n equals 4. Okay, which means that you're going to again have gametes of these individuals that equal 2. So you're going to have 1 chromosome of chromosome 1 and 1 chromosome of chromosome 2. It's important you recognize that in fertilization, we're combining gametes. The 2n numbers, and 2 plus 2 would equal 4 here. So we're combining 2n numbers from each. They're also saying assume the gene regulation on chromosome 1 is not affected by the number of copies of chromosome 1. Yeah, that's usually an issue, okay, when you have more copies. So they're just saying let's just pretend that that doesn't occur and that the genes will act normally if there's more copies. That's important to answer this question. In the 4 resulting viable zygotes that occur when we combine these 4 different or these 4 gametes above. So here comes the question, here comes the action verb, predict. Tell me something in the future that you think is going to happen. Okay, predict the difference in the messenger RNA production from genes on chromosome 1 among the 4 possible zygotes where we're combining, again, normal gamete with each of these errored, I should say, or problematic gametes that have the wrong end number. Okay, so they want to know something about the difference in the messenger RNA production. Well, messenger RNA, of course, is produced when we copy the template strand of DNA, in this case from chromosome 1. So how much chromosome 1 is available? Before we can answer this question, let's add some chromosomes here so we understand what's going on. And by the way, we're adding or we're fertilizing or combining these or fusing the nuclei of the gametes to make a brand new zygote. And because it's an n equals 2, we're going to have a zygote that, or a chromosome that is either going to be dark or clear, and then of course the other smaller one dark or clear. So you get either one, right? So the combinations would only be, there's 4 different combinations, but I'm just going to add a chromosome 1 to each one, and I'll just make it simple by adding a dark one. So that's chromosome 1, and then I'm going to add another chromosome here. I'm going to add a chromosome 1, and again, there'd be different combinations, but I'm just trying to answer the question and keep it understandable. And here comes a smaller one. Here comes chromosome 1, it's bigger, and then here comes a smaller one. Here comes chromosome 1, and here comes the smaller one. Okay, remember, it's 2n equals 2, so I'm only adding 2 chromosomes and 1 from each, right? Again, if you're having trouble with what I'm doing, understand that this could have been the combination in the 2n equals 4 here. They could have been homozygous, okay? The types of homologs from the maternal and the paternal could be the same. So all we're doing is taking just 1 from each, right? The gametes are going to have the number, and I'm only adding that. So when we combine that there, now what do we see? Well, we see with the n+1 original gametes that we got from the non-disjunction that occurred from this cell here, that there is 3 times, and I'll make this a little bit bigger here, there's 3 times the amount of chromosome 1 in the n+1 cells as there is in the n- original cells, right? That's important. So if you have 3 times the amount of DNA from chromosome 1 in the resulting zygotes from mating or fusing with these original gametes that had non-disjunction, we're going to see that there's going to be 3 times the amount of messenger RNA. All right, so again, go back to the question, reread. It's important that we always reread the question. Predict the difference in messenger RNA production from genes on chromosome 1 among the 4 possible zygotes in this pairing or this mating. So I just said the n+1 haploid cells will have 3 times the messenger RNA from chromosome 1 when they become zygotes as compared to the n-1 haploid cells. Now, I don't think you need to write n+1, n-1, but that'd be a great answer if you can recognize that. Anytime you got non-disjunction, it's good to talk about the gametes in that fashion here. So there should be 3 times the amount of, again, messenger RNA made because you have 3 times the amount of the chromosome 1 in half the cells, and then a third less in the other half that result from zygotes from these gametes. And that's my answer there. And I think they could accept a whole different range. I don't think you need to put 3 times as much, but that's a great answer. Looks like you could just say there's going to be more in the cells that have more of the chromosome 1. Maybe that's what they take. It's always interesting to see what where their bar is for a question like this. And then, I didn't mean to confuse you, but to the left here, I just drew a diagram of what this would look like for the gametes if there was no non-disjunction. And again, how these chromosomes line up depend upon what combinations we have, right? It's a 50-50 chance of how these guys could have lined up the flip side, and that would have changed my combinations. But I just wanted to show that, hey, this is a 2n equals 4 in the beginning, and then we have n equals 2, and there's no n plus 1 minus 1 because there's no non-disjunction occurring here. Okay, so on to question 4D. Question 4, part D. Triploidy describes a condition in which an organism has 3 complete sets of chromosomes in each cell. That would be a 3n number, where we usually use a diploid number, which is a 2n. Okay. Provide reasoning to justify. That is our action verb. We're justifying. We have to come up with some reasoning to support something. The claim that most organisms with triploidy cannot produce gametes with chromosomes number typical of the species. Now, what does that mean, chromosome number typical of the species? Well, if your species has a diploid number of 4, we would expect the gametes to be half of that and have n numbers of 2. And what I drew here is a diagram of normal meiosis 1 and 2 without any errors. There's no non-disjunction here. And we can see that we have a total number of 4 chromosomes. Yes, they're duplicated. There are sister chromatins here. There's copies of each chromosome, but there's 4 here total. If you really want to count chromosomes, you count the centromeres, right? So 1, 2, 3, and 4. What's connecting them? Yes, they're duplicated, and they show up duplicated because we want to have them twice. And then when we're done having them twice, we can see that each gamete has the n number, which is half, which is 2, and that would be chromosomes typical of the species. Now with a 3n number, in this case I drew a 3n equals 6. There are 6 chromosomes. Again, the best way to count is count the centromeres. 1, 2, 3, 4, 5, and 6. There's 2 types. There's chromosome 1 and 2, just like we had over here. But there is a total of 6 chromosomes. That's what this equals here. Okay, now when you undergo halving, and there's a couple different ways we can do this, but if I was to halve these and I have them this way, it's not a true halving, right? Because I'm going to just take these 2 and pull off to one side of the cell in anaphase 1 of meiosis. I'm going to take these 4, that's my drawing, and that's what gets me to these 2 daughter cells. So when you think about it, I've got 3 pairs of chromosomes for each set trying to go into 2 daughter cells. And that is never going to work no matter how I slice it one way or the other. I can't get an even number of chromosomes from 3 pairs. Now when I go and now do the next halving, which means I'm going to essentially take this half and make into a gamete. You can see we have a total of 4. And then take this half and make a gamete, a total of 4. Take this half, we're going to have a total of 2. Then this half, take a total of 2. You can see we do not come up with an n equals 3. Very tough to come up with the gametes of 3. Now I separated them, this way into the first round of meiosis, right? But I could have taken these 2. Again, there's different combinations, but here's the problem. How do I split evenly 3 sets of chromosomes? That's the problem here, and that's the real answer here. If I'm going to take an odd number of chromosomes, but there's 3 pairs, how do I split 3 pairs into 2 even numbered daughter cells and then split them into 2 even numbered daughter cells? that is really the answer to the question, is we're taking an odd number of chromosomes and trying to evenly distribute them to an even number of gamete cells, which cannot occur. My illustration here is only showing one of many possible outcomes of uneven segregation. But I'm getting n equals 4, n equals 4, n equals 2, n equals 2. So that's essentially my answer here. So my written answer states the following. Most organisms with triploidy cannot produce gametes with the chromosome number typical of the species due to the odd number of chromosomes. An individual with 3 homologous chromosomes will produce an uneven number of chromosomes in an even number of daughter cells due to the uneven segregation of homologous chromosomes in anaphase 1 of meiosis or anaphase 2 of meiosis. So that's the issue. We're going to have 2 even number daughter cells either after meiosis 1 or after meiosis 2 that give us a total of 4, and we're trying to separate 3 pairs into 2. You just can't do that unless you're going to split chromosomes in half, and that's not going to work. So that is the answer, the best answer I believe, and hope this diagram's helped. On to question 5, part A. Question 5, part A. This is the anole lizard question. So very quickly, this question is really about evolution, right? We have these lizards that live throughout the Caribbean islands in Central South America. I'm just reading this part of the blurb to get you accustomed and acclimated to the theme of this question. This theme is evolution. And how do I know? Well, they're talking about the size of toe pads are genetically determined. That's important. And we see that during storms with high winds, anoles cling to trees with the help of their toe pads. Very important. So their toe pads help them cling to trees. And we're going to have to make the assumption if they can cling to trees and are successful in that clinging or holding on to the trees, then they're going to survive a storm. So we should expect that the storm is going to selectively, in the presence of a storm, that storm is going to be the selective pressure that's going to force the anole lizards to have larger toe pads, because those individuals that had larger toe pads in a population of these lizards would survive better. So what they did was they looked at some data here, and in that data you can clearly see that, again, let's take a look at the legend here, and it's not me. Okay, we can see that where the storms were the greatest intensity, okay, here is where we had the largest average toe pad, and that's moving from 2017 to 2019, so 2 years difference. And because of the huge amount of storms that has transpired between 2017 and 2019, we have a big increase in the size of the toe pads. Remember, these were small in this area, that's the circles, and they became these triangles, which were medium, where there was a large amount of storms. Where there was the greatest amount of storms, you can see the dark colors, that's the diamonds. I know my shapes. So clearly there was a movement from medium, right? From medium, that's the medium size, to large, right? That's the large. And from this position, we went from small to medium. That's the triangles. And so there was an increase in the toe pad size when there was the greatest to the second greatest amount of storm activity. So this is evidence of evolution, as there's a greater amount or greater size of toe pads developing over this time period in the population, which looks like it's in response to these storms. So that's the kind of setup there. And again, did you have to read this? I think you did. I think you needed to acclimate yourself with what this question was about. So here comes part A. Describe the role that changes in abiotic factors have in natural selection. Well, of course, natural selection is a form of adaptive evolution where a population that's diverse enough that already has many different types of alleles present in the population, and where a new group of individuals with alleles that are best suited for this new environmental change, in this case the weather increasing, can now survive long enough to reproduce and increase in frequency of these alleles, which are of course the size of the toe pads. In this case, clearly the weather was the selective pressure. Now they said describe the role that changes in abiotic factors. Now abiotic factors are factors that are non-living, like pH, temperature, well, weather. Weaving your way through this information really helps you understand what this question was about. All right, so again, and again, the action word describe just really talks about the idea that we need to talk about what the abiotic factors are. I think if you just say it's a selective pressure, that's not enough. You can say it's a new environmental force that selects for the alleles that are best suited in that already existing population that get to what? Live long enough to reproduce. And of course, increase that frequency of that allele. All right, so on to part B. Question 5, part B, based on the figure, explain the relationship between the storm intensity and the average toe pad size. So I'm looking at this action verb. It didn't say identify, it didn't say describe, it said explain the relationship between storm intensity and average toe pad size. So if I want to explain that relationship, I need to go a little further based on what the data is showing me. So what is the trend? What is the data showing us? Well, if we look here, I think it's pretty darn clear. And I've done some color coding to help us understand this, but I don't think you needed that. We can see where there is the greatest storm intensity, okay, from 2017 to 2019. There's the largest amount of toe pad size, and the blurb above, okay, as I like to call them blurbs, told us that during storms with high winds, NLs cling to trees with the help of their toe pads. So the toe pads are the bigger pad under their toes, I guess, help them grab onto trees. That is something that physically helps them do that. And if there is a stronger storm or a storm that occurs, if they're able to hang on and weather the storm, sorry for the pun, then they're able to survive long enough to reproduce those larger toe pads to the next generation, and the frequency increases. All right, so we're seeing that medium-sized toe pads went to large, and that's only because the storm intensity was greatest in this area, and we saw smaller toe pads become medium in this area when we looked at the triangles. So there was an increase in the frequency of toe pad size when the amount of storm intensity was the greatest. Where there was least amount of storm intensity, that was in this region here, there was no change in the size of the toe pads. So here we're going from small, right, that's the circle, to a circle here, and that's the least storm activity, and there was no change in the frequency of the toe pad size. So the data is showing that there is definitely a relationship between the size of the toe pads and, of course, in the area with the greatest amount of storm activity. So more storm activity, larger toe pad size. That's the relationship. I didn't say identify the relationship or describe. They want me to explain the relationship. So for me to explain this relationship, I need to talk about a little bit about why the toe pad size is greater in areas with the greatest storm activity. And that's very simple. The toe pad size was greatest where there was the greatest storm activity because the larger toe pads were selected for and helped the lizards live long enough through the storms because they can cling onto the tree and survive long enough to reproduce that allele. It was advantageous to have larger toe pads in stormy weather. They survived more often and were able to weather the storm again by clinging to the tree. So that's really the explanation. So on to part C, number 5, part C. Question number 5, part C. Identify the region on the 2019 map indicated by the number that experienced the least storm intensity. Okay, so we've got ourselves the identify action verb, so we just got to go find it. We don't have to explain it, don't have to evaluate it, don't have to support it, just go find it. So we're looking for the region on the 2019 map that has the least storm intensity indicated by the number. So we're just looking for the number. That's it. Let's go to the map and go find that. So here we are at the map. We're specifically looking at 2019. And we're looking specifically for storm intensity. And we're looking for the least amount of storm intensity. So we're looking for these lighter colors, either no color or the gray, or possibly the stripe if they're all darkened. Okay, so we need to look at the regions. They're looking for which region has the least storm intensity, and that region has numbers, right? So looking for the region number. So region 1, the most northern part of the Caribbean, has this striped color, which is the second highest, greatest storm intensity. And then in region 2, we have the darkened, this is a little bit southeast, okay, part of the Caribbean, and it has the greatest storm intensity. And then region 3, that's a little bit more southeast part of the Caribbean, and it also has a darkened in shade, so that's greatest. So we look at region 4. Hey, that region 4 has colors that are shaded in gray, and gray is lower than the dark and the striped. There is no region that has a shape that's circled with white. So the gray is the least of the 3 colors posted. Therefore, it has the least storm intensity. And they just wanted you to identify the region number. So all you need to put is region 4 or the number 4. And that's the answer. They just asked you to identify. That's your action word. So the answer there is the 4. Region 4 has the lowest storm intensity. On to question 5, part D. Question 5, part D. Based on the information provided, explain how continued differences in storm intensity can apply different selective pressures on anole populations living on the Caribbean islands, eventually leading to speciation. That's an important piece here. We're talking about how evolution, meaning the selective pressures of a continued amount of storm intensities that are differing among different parts of the Caribbean, those selective pressures could lead to a new species of an anole lizard. And the only reason why speciation could occur in that fashion, and I always call this a catchphrase, you hear speciation, think of what? Reproductive isolation. And when you have reproductive isolation between subpopulations, you have yourself a new species. Now, when I think of reproductive isolation, how could the different amounts of, how could I say, intensity of storms in different areas in the Caribbean lead to a new species, or more importantly, lead to populations that could no longer reproduce with each other? So I'm already thinking about a prezygotic or a postzygotic type of mechanism that's going to lead to this. So what I'm thinking is, if you've got greater storm intensity in one area and less than another, that storm intensity obviously could wind up killing lizards. We've talked about this already in previous questions, who have very small toe pads, right? If you have a small toe pad, you can't cling to the tree. This is what they talked about. Well, who else has small toe pads? Well, young lizards that were just born, right, that hatched out of their eggs. They're not going to survive. And if your young don't survive, the next generation doesn't survive. So it makes sense that we can't have young lizards exposed to, that just hatched out of their eggs, exposed to very intense or severe storm weather that would kill them and kill the next generation. So it should make sense that there's a selective pressure on that youth. This is where I'm thinking. So I'm thinking that, well, maybe if there's a few individuals in the subpopulation that is already mating at a different part of the season so that the young are now older with a bigger toe pad who can cling to trees, they would survive long enough to reproduce the next generation. So I'm thinking about temporal isolation, where some lizards are going to start to possibly mate. Okay, if there are already some doing that, right? Already have to have that allele present. But I'm thinking about the idea that they'd have to mate at a different time of the season. And so for me, that's what I'm thinking about in terms of understanding the theme of this question, is that a new population would occur if they could mate at a different part of the season. And if they're mating at a different part of the season, two things occur. Number one, if they're mating far enough away from the hurricane season or the severe weather season, the lizards would be big enough to have bigger toe pads to survive and cling to trees. And second, if they're mating at a different season, this group would now be isolated and not able to mate with the other lizards that are mating later or earlier in the season, depending upon where the severe weather seasons are. So that causes speciation. So for me, that's what I was thinking. And of course, I bet there's other explanations or other answers that would be perfectly fine, but that to me is the obvious one that jumps out. So I said lizards that live in areas of frequent severe weather may select for individuals in a population who mate and reproduce at different time or season of the year that has less severe weather. If this prezygotic barrier occurs, this subpopulation of anoles would be reproductively isolated from the rest of the population, who continue to mate and reproduce at storm conditions or storm seasons, especially in areas where the storm seasons are less severe and their young can survive. So to me, you needed to make sure that your answer spoke to some type of reproductive isolation. All right, I don't think you needed to come out and spell out temporal prezygotic factor or any other. You just need to again explain how these different types of selective pressures are going to. Question 6, part A, based on the box and whisker plot. That's very cool. They gave us a box and whisker plot. Identify the median annual percent change in population size of the unprotected area of region 2. All right, so every question on an FRQ has a theme, and this theme is about this animal called a raptor, and this raptor is decreasing in its population size because of habitat loss, right? Because of human intervention and human living in its same area and taking up a lot of its habitat, and therefore without its habitat, it doesn't have a place to get resources. There's less resources and it's declining. And we have different regions we're looking at. And so this question is about looking at the percent change or the between a protected area that doesn't have any human activity like farming, and then protected areas that don't have any farming, and looking at the percent change over time of the population size of this animal. Any case, in part A, they want you to look at the data, the box and whisker plot, which you should recognize if we've been through enough of these types of data sets, that it measures the median value. It uses the median or the middle number of all the data that's collected, and then there's usually a 50% spread over all the data is collected, and that's the box. And the whiskers, of course, you should recognize as the outliers, the greater number of individuals that are way outside the 50% one way or the other. Okay, any case, they just want us for part A to identify. Everybody loves to identify because you just need a number from the graph. But I'm talking about, again, what's the theme of the question? It really helps to understand how to do every single problem. So they're looking for the protected region, right? This is the region that, of course, there is no human living or farming, and then a certain region. And we're just going to identify the median, right? Where the median value is going to be that middle value. And on a box plot, it's going to be that line in between the box, right? The box is the 50% of the collected data, and the median's right in the middle. All right, and then of course the whiskers, as I just talked about, are the extreme outliers outside of that 50% description of the data. Okay, so we're just going to read. So let's go to the graph or the data set and go get the median annual percent change in the unprotected area of region 2. Let's do that. All right, so here we are at the graph, and we can see that in the legend, I'm not going to say it, okay, not me, but in the legend, okay, we see the protected area is the clear and the unprotected is what we're after, is the shaded. And the question wanted the annual percent or the median annual percent change in population in the unprotected area of region 2. So we're looking for this box plot right here, and the middle value, or this darkened value. You may say, why is it not in the middle? Because remember, it's a 50% range of all the data that's collected. This is the middle value of what was collected, but there could be more individuals that fall in this range as opposed to this range. But in any case, this middle value here is your median. And you probably would have figured that out even if you didn't know what a box plot was, okay? And of course, if we read over, that's going to give us approximately negative 3.0, a negative 3.0 change. And you should recognize that a negative change is we're decreasing. Don't forget the negatives, right? The positive change would be above the 0 line. So you need that negative 3.0. And that's essentially the answer. Whether you want to write a full sentences, you probably don't have time. But the median annual percent change in population in the unprotected area of region 2 is negative 3.0. You could have written negative 3.0 and that would have been the answer. So on to question 6, part B. Question number 6, part B, based on the box and whisker plot again. So we're going to go back to that. If we've been on part A, they want you to identify, that's our action verb. We like that. Just go find the number, okay, of the region and area that experiences the greatest median annual percent change in population size. So in this case, we're identifying the region. What number region is it that experiences the greatest percent change? Now this is was bothersome to some people because, well, what is the greatest? Are we talking about the greatest positive or the greatest negative? They didn't say so the greatest change. So this would have to be the absolute value, right? So the greatest change could be the greatest negative or the greatest positive. Some people read into this to be like, well, if it's the greatest change, that's the largest must be the most positive, and you overthought that. So to me, that part of the question made this a little bit tricky for some to understand what's going on. But if you're looking for the greatest change, they didn't say negative or positive, just the change, then we have to look at the greatest absolute value of that change. So we're going to identify which region has the greatest change, positive or negative. Let's go back to the data set. Okay, so here we are back at the graph of the whisker box plots, and we're looking again for the region and the area that had the greatest median annual percent change. It's positive or negative, the greatest change, right? So we look at the 0 line. That's a really important reference line here. And where is there the greatest change, positive or negative? Well, remember, the theme of this entire question is about the raptors. These are individual organisms that are declining because of humans moving into their habitat, and specifically farming areas. Taking away their habitat and making a farm is lowering their resources, and thus the number of raptors surviving are declining. So you can see that most of these box plots are below 0 because there's a declining population of them. And that's important to recognize that. I think if you were, again, understanding the theme of this question, this becomes an easier answer to easy question to answer. So I'm looking for the greatest change from this baseline 0. And of course, it's going to be the lowest negative value, which is going to be the greatest change. Okay, I should say the greatest negative value here. So if I look at my box blocks, and again, I'm looking at the median values, that's what this is about, I can see the one that has the greatest change or the greatest negative value is region 1, okay, of this shaded box plot, which is the unprotected area, which by the way is negative 4.4. That's my approximate value for that median. So this whisker and box plot is lower for the unprotected area of region 1, and that's essentially the answer. It's the largest change from this point here to point here. Now I know some people got confused. They said, what about the change from the unprotected? No, we're not doing that. I know that a lot of times we compare to controls, and yes, this is the unprotected region, and some people might say, well, from this medium to this medium it's going to be a smaller difference. That's not what they were asking. They were asking for the median annual percent change, and that's just a value from 0 over time. Notice that some of these right here protected areas also decreased from 0. Well, why? Well, because numbers of the raptors are decreasing even in protected areas. There might be some other factor besides farming, right? So, but we're measuring from the 0 line. So all we're seeing is which median was lower. Okay, and of course that is going to be region 1 of the shaded. The shaded, of course, is the unprotected area. So all I needed to say for 6b, the region and area that produced the greatest median annual percent change in population is the unprotected area of region 1. That is all we need to do. Remember, this is the identify action verb. Just go find me the number, region, and the area. Okay, so there it is. So on to number question 6c. Question 6, part C. Researchers hypothesize that if farming is eliminated in region 2, make sure we got this correct, region 2, there will no longer be a decline in raptor population size. Based on the data, that means we have to make sure our answer is based upon that data set. In the box and whisker plot, evaluate, that's our action verb, the researcher's hypothesis. So if I'm going to evaluate a hypothesis, I'm going to see if there's a lot of support, a little bit of support, or no support in their hypothesis. But again, please reread the question, because sometimes you just got to make sure that you understand the theme of the question, and of course understand what the question is about. So we understand the theme is about that we have these we have 3 regions that were been studied. In these regions, we are looking at the range of individuals, and they're basically the median of the population of these raptors in places where the habitat is protected, no farming, and then places where they're unprotected, where there is farming. And we're looking at those differences. But in region 2, they're directing us through this question is directing us to region 2 specifically. And we've got to look at the data to evaluate this hypothesis. And what is it again? They hypothesize that if farming is eliminated, that means if we don't take up and use the area that was once the habitat of the raptors, that area would support more raptors. There would be more of those individual species living, and the population would be bigger. So for me, we need to look at the region 2 and look at the protected area, because we're saying that the unprotected area was farming, and now we're going to, and we know that the farming is taking up their habitat. Let's take a look at the protected area. Is there a difference between the protected area that in region 2, because they're specifying that in region 2, is there a difference between the protected area, okay, value for the median population size of the raptors compared to the area that's being farmed and unprotected? Because that's really going to either support or not support the hypothesis. So let's go to the data set and see. Okay, so here we are at the whisker and box plots for this data set for the raptors, and their population mostly decline, as we can see. Now, if you look specifically, the question was about region 2. And in region 2, we're going to compare the unprotected area. That's the area where there's farming, and we see there's approximately a negative 3.0 median annual percent change. It's decreased. It's a negative 3.0. We use that value for another question. But now let's take a look and compare that with the box plot where it's protected, because remember, the researchers hypothesized that if we didn't have any farming, that the population would be greater. There would no longer be a decline. We're seeing a decline. Negative 3.0 is a decline. So what does it look like for the protected area? When we take a look at the box plot of the protected area, again for region 2, that's where they're pointing us to, we can see that the median value is 0%. There is no change. There's no positive, but more importantly, there's no negative change. There's no decrease in population of the raptors when the area is protected, right? This median line is at the 0 mark. There's no change in population. There's no decrease in the population when we protect the land in region 2. So this supports the researchers' hypothesis that there will not be any decline in population if we protect the land, because there is a whisker box plot that has no change when it's protected. And that's essentially the answer. Okay, and again, using the values of this table, you need to express that. The words that I used, I said the median annual percent change in raptor population in the protected area of region 2 is 0%. A 0% median annual percent change indicates that an area with no loss of habitat and with no farming occurring does not have a negative percent change or a declining population, while the unprotected area with farming has a negative 3.0% change. Many ways to say that, but you needed to go after that. And yes, we're supporting that. Now, I know a couple of people may have thought that because the box plots are overlapping, that there is no significant difference. Careful, careful, careful. That part, I don't like how they set this problem up, because many people didn't even look there, said there's no difference because the box plots are overlapping. These are not error bars. These are whiskers, and these are not error bars also. So we don't look at significance by overlapping box plots. So there really is no significance. This is descriptive data. This is there is no p-value, there's no probability, so we don't look at whisker box plots for significance when they're middle 50s, by the way, they call these the middle 50s overlap, right? So yes, there is some overlapping in their middle 50s, but that's not any part of significance in descriptive data. Descriptive data is standard deviation. It's all of the values that you measured. Remember, standard error of the mean is not descriptive data. That is the reliability factor that gives you confidence in your descriptive data. So this is not something we look at. So unfortunately, I don't think that the College Board should have overlap these. I know that some people confuse that. All right, so I just making the mention of that again, overlapping box plots isn't a value of significance or confidence. All right, so on to question 6, part D. Question number 6, part D, based on the information provided. Okay, so that's based upon the blurb of information they gave you, the theme of the question about these raptors that have a declining population, mostly in areas that are being farmed, where their habitat is being taken away. And of course, in some instances, we have the population is not decreasing in region 2 specifically. That we just talked about that in part C, where their areas are being protected. Explain. Okay, so that's a little more work and explanation here. Why ecosystem resilience? This is a catchphrase, my friends. Resilience is a word that pops up a lot when we try to explain why ecosystems can bounce back when there's a change or some kind of stress added to an ecosystem, like an invasive species, when there's a great amount of genetic diversity. So in this case, they're saying why the ecosystem resilience in the unprotected areas where the humans are taking up the habitat of the raptors may decline over time. So you're trying to explain why the resilience, okay, is going to decrease, is really equal to the idea that the stability of the ecosystem is going to decrease. And the reason why it's going to become less stable probably is due to a lower genetic diversity. There's somehow the ecosystem is going to be less genetically diverse, which means energy can't travel up from the producers to the consumers as well. And there's going to be a breakdown and possible collapse maybe of this ecosystem. Resilience is a word that pops up in this curriculum a lot, and they're just kind of using this word backwards. I've seen the word resilient used as an answer to many questions regarding ecosystems and their amount of genetic diversity. So stability is going to be dropping, and so is genetic diversity. This ecosystem will have less ability to bounce back somehow for some reason in the unprotected areas. Well, what's the theme of this question? The theme is the unprotected areas. The raptors their habitat is being taken up by humans and used for farming, so they have less resources to live. So what about the loss of raptors? Well, you could say that, well, losing and possibly losing a species like a raptor, may actually lose that species in these different areas, may have a negative effect on the complete ecosystem and lower it, and it may not. But usually losing a species will make an ecosystem be less stable for sure than it would if it had it. But I think they're after more here, and they definitely are, because they told you something in that little piece of information that started this question. Let's go there, and I think that's the key to this problem. So yes, the beginning of the problem here, they say many raptors in Africa are top predators and keystone species. We should know that a keystone species is a species that has its niche in the ecosystem, supports many other niches or jobs of the ecosystem. If a keystone species is to disappear, and again, it's based upon the information or the data here, okay? So if you look at the data, we can see that there is clearly a decline in all three regions using the box plots, the median values in the unprotected areas. There's going to be a continual negative decrease in population side year after year in these three regions. Then what we're going to have is possibly a loss of these raptor species in these ecosystems. And, par that with the fact that they're keystone species, which means they support many other important species that deliver the energy up through the trophic levels. Hey, that's an important piece of information you needed to know. And when you think about it, boy, you just took the multiple choice section of the test for 90 minutes, and now you're at the end of the last 90 minutes trying to get all these answers in, and here we have the last question of the test. Expects you to go back and make sure you read that first line. That makes this a challenging test at times, and I've seen this a couple of different tests where you needed to make sure you understood that. So I think because they gave you that piece of information, you needed to answer this question along the lines that, well, because we're going to lose a keystone species, the resilience is going to decrease because possibly the loss and lowered amount of the keystone species is going to make the ecosystem less stable, and therefore lose other organisms that support the ecosystem and drop the genetic diversity even more. So that's what this was about. The fact that based on the information provided, I think the provided part was about going into the first line that, hey, it's a keystone species, and understanding keystone species are ones that if lost, the entire ecosystem collapses. Right? So my written answer is the ecosystem resilience in unproductive areas may decline due to possible loss of raptor population if habitat loss continues. The raptor is a keystone species. That's where I got the information from provided. And with a declining raptor population, the ecosystem will become less stable and may collapse. I use the word collapse because when you lose an ecosystem, ecosystems break down, leading to loss of many other species and a lower genetic diversity. Okay, so yeah, they can't bounce back because you may lose many organisms. The whole ecosystem will possibly collapse if we keep losing more and more of the keystone species, in this case the raptors, that's supporting other species. All right, so I hope these explanations helped in a big way. Good luck on your AP or understanding the curriculum.